So this lecture covers Chapter 6, Sex Matters, in your textbook. And just before we proceed into this chapter, just want to remind you what we've been looking at for Exam 2. So Chapter 4, we started at looking at Mendelian inheritance. So remember, those were situations where we had a complete dominance and recessive inheritance, where just one gene was influencing a phenotype. So those were our simplest scenarios. However, we know that there aren't many phenotypes that are actually inherited in this fashion. So we went on to cover a wide range of phenomena in Chapter 5, in that Beyond Mendel chapter, that differed with regards to inheritance patterns. So all these different phenomena had, rather than our expected 1 to 2 to 1 genotypic ratios when we crossed heterozygotes, they varied with regards to what we'd see. Or instead of our expected 3 to 1 phenotypic ratios, where as long as we had one dominant allele, we would see the same phenotype, these phenomena varied. Very often we would see a new phenotypic class within our heterozygotes, for example. Some of the other factors involved multiple genes or more than just 2 alleles that we covered in simple Mendelian inheritance, or interactions between genes and the environment. So the other part of Chapter 5 was studying the inheritance of mitochondrial DNA. So we explored how the genome of mitochondria is maternally inherited and therefore altered our Mendelian expectations. So with regards to Mendelian inheritance and all the phenomena we covered in Chapter 5, we dealt strictly with autosomes. So chromosomes 1 through 22 were involved in these different factors. In this chapter, we're going to focus solely on the inheritance of sex chromosomes. So the different inheritance patterns that we see due to dominance or recessive genes that are linked or tied to these sex chromosomes. Particularly, we're going to focus on the X, but we're also going to explore a few other conditions in which phenotypes vary between males and females. So we're going to focus on our X chromosome mainly, but our Y chromosome as well. So Chapter 6 starts out talking about what we really mean biologically when we're talking about the sex of an individual. So maleness or femaleness, for lack of another term to define these attributes, are both determined at conception. And that is due to the presence of the sex chromosomes that are passed on via the male and female gametes. So just like the rest of our chromosomes, we have a homologous pair of these chromosomes. And the genes on those chromosomes are expressed or transcribed and translated during development within the womb. And their expression triggers the formation of different cell lines that will ultimately give rise to production of gametes and production of hormones and production of the different pathways related to male or female reproductive systems. So all of those are on the homologous chromosomes, and in this case, we've already covered this, but remember that males have one X and one Y chromosome, while females always inherit 2 copies of the X chromosome. So that's at the genetic level and related solely to genes on those chromosomes. So another level of sexual identity comes from the control that hormones exert on development. So after an individual is born, a lot of sexual maturity happens or starts to happen at puberty, and that's due to the different hormones that are being expressed. And obviously that comes back down to the underlying genetics, but we don't see those hormones really kick in until puberty and later on. So finally, another aspect that influences the sex of an individual, and really I like to use the term gender here. This comes down to gender identity, how an individual views themselves with respect to their sexual identity. And that relates to both psychological and sociological components that influence sexual feelings. So this can sometimes be completely different than what an individual actually inherits in terms of the genes influencing their sex. So just a reminder that we have a biological level that influences things, but there's another level that influences and contributes to gender identity. So let's explore what happens during embryonic development that contributes to the biological aspect of sexual identity. So differences between the sexes actually don't become apparent until the 9th week of prenatal development. And that's because starting finally in the 5th week, we start to see some precursors to the gonads and the duct systems. So remember that in the very beginning we have our sperm and egg, they unite. That's our zygote. The zygote undergoes lots and lots of mitotic division, so we have more cells to work with. Those cells finally start down some developmental pathways so they can become specialized, and they do that by expressing different subsets of genes via transcription and translation. So during the 5th week of development, the cells that are ultimately going to give rise to the gonads and other reproductive tissues start to form into these different precursors. So we've got gonads, but they are unspecialized. So even though they're going to go on to become ovaries or testes, at this moment in time, at the 5th week, they look completely identical to one another. The developing individual has just one set, and if we were to compare an individual that was ultimately going to become male versus one that became female, we wouldn't be able to tell the difference at this point. We also have 2 sets of reproductive ducts. So in every individual at this point, they have the same 2 sets of ducts. We have a Müllerian duct, which we consider to be female specific, because we will see that it will go on to become or give rise to the uterine tube. And we also have a Wolffian duct, and that's the male specific duct. It's going to go on to become the vas deferens of the male. But at this point, every individual has both. So the opportunity for that individual biologically to become male or female lies within the genes on the Y chromosome. So this is all about the fact that really males have a Y chromosome, females don't. But it's because of a specific gene on the chromosome that triggers the production of hormones that ultimately steer development towards one route or the other. So this SRY gene, if present, and we call this SRY gene the sex-determining region of the Y chromosome, because ultimately it's just due to the presence of this gene. So if this gene is present, and during week 6, it is expressed, it is transcribed and translated. When that happens, hormones are produced. And if those SRY hormones are produced, that triggers the individual to go along the male pathway. So what that signals is for the deterioration of the Müllerian ducts, or those female precursors, and the full development of our Wolffian ducts into our vas deferens. If that SRY gene is absent, so those hormones simply aren't produced, that causes our individual to move along the female developmental pathway. And on the next slide, we'll see an image of this. So this is figure 6.2 from your text, and it shows visibly these different pathways of development that I just described on the previous slide in a verbal approach. So during the 5th week of development, we finally start to see the appearance of these reproductive structures. So we have our paired gonads, but they're unspecialized. So they haven't traveled down any other developmental pathways to look any different from one another. But we also have 2 sets of ducts. So we have the Wolffian ducts, we have the Müllerian ducts. So as long as our SRY gene on the Y chromosome is present, at week 6, we see 2 things happen. The gonads start to become specialized. So due to the expression of specific hormones produced when this SRY gene is turned on, is transcribed and translated, we now see these unspecialized gonads turn into our testes. We also see that the presence of these hormones actually signal for the degeneration or the breakdown, disappearance of that Müllerian duct. So our Wolffian duct is going to be the one that stays within the body and goes on to become more developed, but we lose the other one. And there's more steps that need to happen before this duct, as you can see in this pathway, ultimately becomes the vas deferens. If there simply is no SRY gene present, so that means we don't have a Y chromosome, we've got copies of our X chromosomes, the hormones associated with this transformation just aren't going to be produced. And if they're not produced, we can no longer see the development of that Wolffian duct. So it starts to break down. There are no hormones present to support its further development. So what happens in that case, the Müllerian duct is the one that goes on to respond to the different expression of genes and ultimately be transformed into our uterine duct. The other thing we see is that our gonads, if the hormones triggered by the SRY gene are absent, those gonads go on and become specialized as ovaries. So biologically speaking, it's the action of these sex chromosomes that are ultimately going to determine, and I don't like that I have gender here, just sexual identity from the biological perspective. So human males are what we call the hetero, meaning different, gametic sex. And that's because they have these two different sex chromosomes. They still inherit one X from their mother. They also inherit the Y chromosome from their father. Human females are what we refer to as the homogametic sex. So that means they have two of the same sex chromosomes. In this case, they inherit one X chromosome from each of their parents. So note that in other species, sex can actually be determined in different ways. So for example, in birds and snakes, males are actually the homogametic sex. So we're dealing with even different names for their chromosomes here, the ZW system. But what you notice is that males actually have, in that case, the two identical sex chromosomes, two Zs, while females are the heterogametic sex. They still have a Z, but now we see a different sex chromosome, which is called the W. Let's look a little bit more in depth at these sex chromosomes, so the X and Y chromosome. So they really differ. The most obvious way they differ is in size. And because of their size differences, that means that they have different capacities in terms of how many genes that they hold. So the X chromosome is much larger, and because of its large size, actually contains more than 1,500 different genes. So remember, it's still our homolog when we have our different pairs of chromosomes. Our sex chromosome pair, we can see two X's, we can see an X or a Y. So those are our homologs. But our X chromosome is larger and has many more genes. Our Y chromosome is much shorter and it looks very unique in terms of its structure. It's how it gets its name. It has only 231 protein encoding genes. Many of the genetic segments of these genes are actually palindromes, so meaning they read the same forward and back, and that can actually destabilize our DNA. And when this happens, when the DNA becomes destabilized, different parts of the Y chromosome attract one another, and they actually form different loops and end up deleting sections of the DNA. And scientists have discovered that that action may actually result in male infertility. And we've already talked about the most important one of these genes, our SRY gene, because it influences gonadal and duct development. So here we have a schematic of the Y chromosome. Let's take a look at its unique anatomy. So we've already seen on the previous image that our Y chromosome has a short arm up on top and a long arm on the bottom. At the end of both of these arms are these specific regions. One is called par 1 at the top, and the other at the bottom is called par 2. And these regions we refer to as pseudo-autosomal. And they make up only a very small proportion of the chromosome, only about 5%. And they're unique because they actually include 63 genes that encode proteins for function in both sexes. So that means we see these same exact genes on our X chromosome. So that's why the X and Y, even though they look different, are referred to as homologs, because they have homologous genes, granted just a few of them, but this enables them to cross over during prophase 1 of meiosis 1 and swap genes. So they're able to recombine and share information because it's the same genes found in these regions. So the majority of the chromosomes, about 95% of it, does not recombine with the X. And we refer to this as the male-specific or non-recombining region, or MSY. So this is where we see the majority of our 231 genes. So this whole region of the chromosome actually contains many genes that relate to sperm production and therefore influence fertility. So our SRY gene produces hormones related to sperm production. We have this AZF gene, which also influences production. We also have this gene here, unnamed, but it's going to contribute proteins and hormones that influence sperm development. So that would make sense. A lot of genes that influence gametes directly. So the MSY region also has some sequences that are identical to the X, but they aren't protein encoding genes. So it's why we don't see any swapping over. We've also got about 20% of this MSY region that are similar to X chromosome sequences, and the idea here is that they may be remnants of that ancient autosome that actually ended up specializing to become our X. So again, it kind of relates to the homologous nature or similar nature of these two chromosomes that are our sex chromosomes. And the rest of the MSY is full of these palindromes, which I've already mentioned. So these repeated sequences that read the same forward and back and can actually swap with one another and break off. And it's one of the reasons, these palindromes is one of the reasons why sometimes we see fewer genes or more genes in this MSY region when comparing individual males. So I've mentioned the importance of the SRY gene several times now in terms of going on to influence development of male structures and other aspects of maleness. So how does this work? Well, the SRY gene actually encodes a specific protein called a transcription factor. And we've already learned about transcription factors. So remember, transcription factors are important because they bind with other binding proteins at gene promoters. And when they bind all together, they essentially form a pocket that allows for RNA polymerase to come in and begin making an mRNA transcript. So what this means ultimately is that these transcription factor proteins control expression of other genes. So the SRY gene encodes the instructions for a specific protein called a transcription factor that then controls the expression of other genes. And those other genes do a couple of things. So the expression of these other genes do multiple things. So one is they produce hormones that stimulate male development. And we've already covered how one of the roles of these hormones is to specialize the duct system. So some of the hormones that are produced by the developing testes secrete what's called an anti-Müllerian hormone. And that anti-Müllerian hormone, when present, actively destroys the female structure. So it destroys those Müllerian ducts, and if the Müllerian ducts can't be around, then all that's left are the Wolffian duct system to go on and to develop. So once we've gotten rid of the Müllerian ducts, we have the production of testosterone and dihydrotestosterone, which is called DHT. Clearly, I am not very good at pronouncing that. So they're both secreted, and the presence of them not only goes to fortify or further develop the vas deferens so it can take its proper shape and travel through the pathways that it needs to go and become fully developed so that it can function, but it also triggers the further development of the testes, the epididymis, all of the other structures and glands that are associated with the male reproductive system. So your textbook provides an overview of things that can go wrong with male development. So obviously, we are dealing with a gene in this situation. So any types of mutations in this gene that encode different amino acids than were originally encoded can cause problems with any of these pathways. So there are many things that could potentially go wrong because we've got the SRY gene serves as a transcription factor that then goes on to influence the expression of other things. So we can have the potential for multiple places in these pathways for things to go wrong. And if you're curious about learning more about that, you can look at figure 6.4 in the text, but I'm not going to specifically cover any of those things in this lecture or require that for you on the exam. So as I've mentioned a few times, sexual identity does not relate solely to the genes and chromosomes that we inherit. And I think your textbook does a nice job of summarizing the different levels or aspects that contribute to sexual identity. So table 6.1 just provides an overview looking at the different layers involved or different aspects involved, the events that relate to the aspect, and the timing in terms of development when these things occur. So as we've been covering on the previous slide, at the most basic level, sexual identity is determined by our chromosomes and the genes on them. So the Y chromosome is what carries that SRY gene that contributes to maleness. So right after fertilization, male versus female is defined by the chromosomes that are present. So XY is a male and XX is a female, biologically speaking, and that happens very early on, right away as that zygote is formed. So sex as it relates to the gonads, the reproductive organs, remember that we finally get an undifferentiated structure because both males and females have that same unspecified gonad, and it doesn't start to develop into a testes for males or ovary for females until about 6 weeks after fertilization. So finally we will see the male precursor to the testes and the ovary for a female. So phenotypically, in terms of development externally and internally of other reproductive structures, continues as male or female in response to the hormones that are being expressed on either our X or Y chromosomes. And this happens about 8 weeks after fertilization, when the tubal structures are predetermined finally to specialize and be present as either the vas deferens or our oviduct. But it continues through puberty, as these hormones are really critical to the reproductive cycles of both sexes. And we've learned about that in the meiosis chapter. So the next layer is gender identity, and that relates to the feelings that a person has, whether or not they identify with being male or female. And those identifying feelings or relationships with oneself starts typically at childhood, but it could potentially start earlier for all we know. So finally, we get to sexual orientation. So you can have a certain gender identity, but be attracted to a different sex from your identity, or the same sex, for instance. So this layer of orientation relates solely to the attraction of either the same or opposite sex. And again, as gender identity, this typically starts from childhood. So there's a lot going on in terms of the biological components of sexual identity, the emotional components, and sociological components that contribute to sexual identity. So with a lot of things, it's not just due to our genes. Understanding how sex chromosomes are inherited is important when we're working through problems where we want to determine or predict our phenotypic and genotypic ratios in our offspring. So we've already learned that biologically, a female is the homogametic sex, so she has 2 copies of the X chromosome, while a male is heterogametic sex, has 1 copy of the X and 1 of the Y. So just reminding you that all of our genes come in pairs, and that's because we have 2 sets of genetic information stored on these homologous chromosomes. So when passing on these chromosomes to the next generation, these homologs are separated during the first round of meiosis, and then the sister chromatids that make them up are split up again. So for a female, she has a 100% chance of passing on an X chromosome to her offspring. And that's because it's the only possibility that she has. For males, there's a 50% probability that he will pass on an X chromosome in his sperm, and a 50% probability that he will pass on a Y chromosome in the sperm. So now we turn to completing our Punnett square, where we combine these chromosomes, and when dealing with alleles, the alleles that would be on them, but in this case we're just showing how the chromosomes are passed on. And we can see that 2 out of 4 of the offspring, or 50%, are females, and 50% of the offspring are males. So there's a 50% chance that each time a zygote is formed, that it will be male, 50% chance it will be female. So the next few slides are going to work through problems where we have genes located on either the X or the Y chromosome. So they're linked to that chromosome. So I just want to point out before I move from this slide, the X-linked inheritance patterns all come down to the alleles on the X. And in this case, we can see how, if I denote a mutation by a letter M, if a female is a carrier for a mutation, so in that case, we would have a recessive disorder, only one copy of our allele is faulty, that's why we say carrier. That mutant is going to be passed on half of the time. So there's a 50% chance that either her son or her daughter will inherit that mutation. So the X-linked traits are always passed on this X chromosome from a mother to son, or from either parent to a daughter. Because we can see, if I place this mutation on the male's sperm, he passes on one X to his daughter. So in that case, he will always pass on the mutation to his daughter. There's a 100% chance that he will do so because one of the daughter's copies of the X chromosomes comes from her father. So now that we understand how these chromosomes are passed on, let's talk about the phenotypes that these chromosomes influence. So the chromosomes themselves have genes, and if those genes are related to specific traits or disorders, then we refer to them as sex-linked traits because they're passed on via the sex chromosomes. And it all depends on which of the chromosomes that the genes are on. So if these genes underlying a particular disorder or phenotype are located only on the Y chromosome, we call those Y-linked. These types of disorders or traits are extremely rare, and that's because when you think about the anatomy of our Y chromosome, remember it's really small. It only has 230 total protein encoding genes, and remember that 63 of those, the ones at the very tips, are homologs. So they are shared in common with the X chromosome. So because of that, they're not just linked with the Y. The rest of the genetic information within the main portion of our Y chromosome contains other stuff like those palindromes and other things that are kind of ancestral copies. So when it comes down to it, really there's only about 20 genes that are unique in having some role of protein function that we only see in males. So that explains why these conditions are very rare. They are also transmitted only from males to males. So father's going to pass it on to his son if he has a mutation, but not to his daughters. And that's because he's only passing on the Y chromosome to his sons. So we don't see affected females when we're looking at pedigrees related to Y-linked traits. So currently, some of the identified Y-linked traits, so phenotypes that are influenced solely by genes on the Y chromosome, relate to infertility. We've discussed that a bit because 3 of those main genes control sperm production and function. But obviously those can't be transmitted because if they're affecting or leading to infertility, there's no way that that male can pass on the genetic information. And interestingly, the other genes on the Y chromosome that have been attributed to certain disorder actually have been inserted there, so they come from other chromosomes. Weirdly enough, they recombine with the Y chromosome, like Y-linked deafness, which scientists used to think that was solely due to the genes on the Y chromosome, but it's not. It comes from a different chromosome. So more work needs to be done here. So we refer to disorders that are inherited based on genes present on the X chromosome as X-linked traits or disorders. So our X chromosome, because it's much larger and contains overall more genes than our Y chromosome, is responsible for most of these conditions. And when thinking about the X chromosome, it accounts for 4% of all the genes in our genome. But interestingly, it accounts for 10% of all Mendelian disorders. So there's a disproportionate number of X-linked genes that actually cause illness when they are mutants. Genes on the X chromosome also have different patterns of expression in males versus females. So we can use that information when we're working through inheritance problems. So the reason we see different patterns of inheritance or patterns of expression is because females have 2 X chromosomes and males only have 1. So when we work through the possible genotypes in a scenario, and in this scenario we're going to be working with an X-linked recessive condition. That's because I see the term carrier, so I know we're dealing with a recessive disorder. Females have 3 possible genotypes. So it's exactly the same like we've been working through with all of our autosomal conditions when we're dealing with just one gene. So because she has 2 copies of the X chromosome, she also has 2 of the alleles. So if she has 2 of the normal functioning alleles, we refer to this as homo because it's the same. Zygous, there's a spelling mistake there, sorry. Wild type female. If she has 1 mutant allele, we refer to her as heterozygous, and therefore she's a carrier. Finally, there's a possibility that because there are 2 X chromosomes, she has 2 of these mutant copies. And in that case, we use the term homozygous, and then we would state mutant female. For the males, there are only 2 genotypic possibilities. And that's because males only inherit 1 copy of their X chromosome, and therefore 1 copy of whatever allele is linked on that X chromosome. is found on the X chromosome. So in other words, there is no extra backup copy of that allele to mask the expression. Whatever they inherit on their X chromosome from their mother, that is what will be expressed. So in this case, the two potential genotypes, in both cases, we're going to use the term hemi. And that's because we're only dealing with half, right, the information. They only have one copy of the X chromosome instead of two. So if they inherit a functioning copy of the allele, they're hemizygous wild type. But if they inherit a mutation on that X chromosome, they're hemizygous mutant male. And in that case, they're going to have a phenotype related to that mutation, no matter what, whether it's dominant or whether it's recessive in nature, because they only get one copy. So I'm just showing you at the bottom here, I'm using this M to denote that the allele at the gene of interest that we're working through is representing the mutant. So there's a mutation that's causing a problem and therefore being associated with a particular phenotype. So this slide summarizes what to look for when examining the transmission of an X-linked recessive trait. So in this case, the mutation is going to be a recessive allele, and because we're dealing with X linkage, we find this allele on the X chromosome. So to denote that as a genotype, and in this case I'm just showing half of the genotype, we use the lowercase x with a superscript m to denote the mutation. So these types of conditions are always expressed in the male when the mother passes on an X chromosome that carries the mutation. We've already discussed how men only have one X chromosome, so whatever they inherit, that is what's expressed. With regards to females, because this is a recessive condition, and remember that females have two copies of the X chromosome. So it's going to be expressed when two copies are present, because that's the definition of how a recessive allele works. But it may also be expressed when she only inherits one copy. So always expressed in a female homozygote, but rarely in a heterozygote. And the reason for that we're going to cover in more detail in a few slides. But this is because of X inactivation. So it deals with the fact that one of the X chromosomes is randomly going to be turned off. And in the heterozygous condition, if it's the normal functioning copy, we're going to see the condition or symptoms associated with the condition in the female. So when dealing with how these conditions are passed on, if we have an affected son, what that means is the mutation was either passed on from a heterozygous or carrier mother. Here you can see she has one copy of the mutation, or a homozygous mother. And depending on the condition, if a heterozygous mother is passing that condition on to her offspring, her son, if there's a 50% chance that she'll pass on that mutation on the X chromosome. On the other hand, if the mother is homozygous with regards to the condition, there's a 100% chance that she is going to have an affected son. So notice we don't deal with the father in this situation, because the father is only going to pass on his Y chromosome to his sons, not the X's. On the other hand, if we're dealing with an affected female, she absolutely has to have an affected father because she needs 2 copies, sometimes rarely 1 copy, of the mutation on the X chromosomes. And because she gets one of her X's from her father, he has to have the mutation. And her mother can either be affected or a heterozygote. So in either scenario, the mother is able to pass on the mutation to a daughter. Here we have a list of some examples of X-linked recessive phenotypes. And these conditions are not lethal. And therefore, a man that has a single copy of a mutation is typically healthy enough to transmit it on to his offspring. So even though he suffers from symptoms associated with the disorder, they aren't extreme enough to prevent him from passing it on. So these 3 examples are ichthyosis, which we'll cover in more detail on the next slide, but this is a deficiency of a particular enzyme that removes cholesterol from skin. Color blindness, which you likely all have heard of, is an inability to distinguish between colors. So the most common example is red and green color blindness, but there are other color combos that may be difficult for individuals to distinguish between. And your textbook provides a really nice overview of this in the Clinical Connection 6.1. Finally, a really well-known, kind of famous example of an X-linked recessive condition is hemophilia. And this condition is a disorder related to blood clotting, and we're going to cover that in more detail in a few slides as well. So here we have an individual that's presenting with ichthyosis. So our skin is comprised of many, many layers. In particular, the epidermis of our skin is made up of stratified squamous epithelium, and there are up to 50 layers of these squamous cells. The living cells are located at the very bottom of the layer, and as they age and die, those are at the top. And so this is what allows for the protective nature of our skin as an organ and prevents moisture loss. With this condition, however, there's an enzyme deficiency. And what that enzyme deficiency does is blocks the removal of cholesterol from these dead skin cells. Consequently, that very uppermost layer cannot peel away as it normally does. So it simply can't be shed. If it can't be shed, it's going to remain in position and stack up over time. Because of that, the skin appears brown and scaly. So when we look at a pedigree of this condition, what we see is an affected father, and in this case, because we're dealing with 3 generations, this is the grandfather in the scenario, and that is who we're looking at in this individual picture. So how can this condition be passed on when we have only affected males? So we have an affected grandfather and his grandson being affected. But we lose sight of this phenotype. It appears to be missing. It skips a generation. So the only way that this is possible is for this mother to be a carrier. So she's heterozygous for the condition. She doesn't show it, but she still has a 50% chance of passing on that mutation to a son. And in this case, if she does that, he is going to show the same phenotype as his grandfather because he doesn't have an extra copy of the X chromosome to mask anything else that's present. So you can see that Only males are affected in this small pedigree, and females are carriers. So figure 6.7 from your textbook shows a very extensive pedigree. So not limited as we saw in the previous example. This is very well tracked across royal families. And this is hemophilia B. So it's another X-linked recessive condition. It's also referred to as Christmas disease or factor IX deficiency. So biochemically, for our blood to actually clot when an injury is incurred, it involves a large number of factors. So it's sort of like a chain reaction for a number of proteins to be produced for signals to be passed along and steps to be taken by the blood. If one of those factors is missing, and this is the case for hemophilia B, problems result. So the blood fails to clot, and the individual has a longer bleeding time than someone who has all functioning factors. So the upper left-hand corner shows you a Punnett square involving the crosses of individuals. And I'm not going to go over this right here because I'm going to do so in detail on the next slide. However, I want you to take a look at this pedigree, and let's look for some things that really stand out in terms of how this inheritance pattern appears over generations of time. So typically what we see with these conditions is that there's no previous family history of them. So we're dealing with a recessive condition, and that can skip generations. But to distinguish that the condition is carried on the X chromosome and not an autosome, it's a couple features that stand out. So one, these conditions are passed on via heterozygous or carrier females. So all of the females that we see in this pedigree through all of the generations, there's no female that is actually showing two copies of the condition. They are all carriers in these cases. And some of those females may be actually affected by the condition due to X inactivation. But for this case, we absolutely know they have to be carriers, because there has to be a way for that mutation to be passed on to the sons. So the other big key that stands out in this pedigree is that we have a large number of affected males. No carriers, all affected. And we see this in every single generation across this pedigree. So that's a signal that the condition almost looks like it's dominant, in fact, right? We're not seeing a carrier, we're seeing it present in every single generation. But it's not a dominant condition because we don't see that same inheritance patterns in females. So what that tells us is that this condition has to be carried on the X chromosome. And here's our mutation. Okay, so the males are hemizygous for these conditions, so they only inherit one X chromosome. Whatever they inherit is what's going to be expressed. So hemophilia was actually passed on between royal families, and that's because historically members of different royal families would marry one another. So we see hemophilia present in the Spanish royal family, German royal family, and the Russian royal family. And although it was present in the English royal family, historically, it no longer is. So let's walk through creating the Punnett square for X-linked recessive traits. So as I've already mentioned, We typically see these disorders being passed down initially from a carrier mother and an unaffected father. So if we start out like that, we know our mother is a carrier. So her genotype, she has 1X, that in this case you can see is carrying the dominant or functioning allele, represented by an H. And on her other X chromosome, she carries a mutation. So this is represented by our lowercase h because our condition is recessive and X-linked. So for our father, he is unaffected. So half of the genotype is an X chromosome with that normal functioning copy of the H allele, and the other half of his genotype is a Y. So let's cross all of these gametes, and you can see there's a 50% chance for both parents that they're producing these two different gametes. Okay, so in our top leftmost square, we pass on both of these normal functioning H alleles on the X chromosome. So we end up with a female, and in this case, she would be normal functioning because she has the wild type alleles for this particular gene of interest. However, in this square, we have another female, so a daughter, that actually is heterozygous. So she ends up inheriting the mutated H allele, recessive allele, from her mother. So now she is also a carrier. So you can see here that if we already know that we have a daughter, there's a 50% chance that she's going to be homozygous dominant and not be affected by the condition and not carry on the mutation. We also have a 50% chance that she will be a carrier and inherit that mutation from her mother. So let's complete the bottom part of the square. We have a normal functioning allele being passed on, and now we have a Y. So in this case, we have a son, and he inherits the normal wild type allele from his mother, so everything is perfect functioning. In this bottom right corner, however, the mother is passing on that mutation to her son. He has no other copy of an X chromosome to mask its effect, and therefore this son is affected. So we can see here that we have a 50% chance when dealing with sons that they will be normal, and the other 50% chance that they will be affected. So here, all of these points done out in text for you kind of summarizes what I've gone over with the Punnett square. So we can see first, so a carrier mother has a 50% chance in passing on that recessive mutant allele to her offspring, right? So that's everything that's happening in this right side of the Punnett square. So 2 out of the total 4 possibilities, right? 2 out of our 4 possibilities, we see that recessive allele present in the offspring. So that's a 50% chance. An unaffected father will only pass on his dominant allele, so the wild type, normal functioning, to his daughter, and a Y chromosome to his sons. So we see this normal wild type X being passed on from father to daughter in both cases. So he's always going to pass on a normal functioning allele. So no daughters in this case, looking at the top line of the Punnett square, will express the phenotype, but they do have a 50% chance of being a carrier. The only side note on this is when we cover X and activation in a few slides, it is possible that you can see a carrier female show at least some symptoms associated with X-linked recessive trait. But if I tell you on the exam, ignore X and activation, no daughters will express the phenotype. Because we see here 50% of our daughters are normal, 50% are carriers or heterozygous, neither affected. Sons, however, have a 50% chance of being normal, so no symptoms showing with respect to the phenotype, and a 50% chance of being affected by the condition. So now we're going to switch gears and look at X-linked dominant inheritance patterns. So these types of conditions and traits are actually quite rare, and we'll discuss why in a few minutes. And the gene expression differs between the sexes. And that comes down to the fact, again, that females have 2 copies of the X chromosomes, but males only have 1. So here we see a summary of what we're actually looking for in terms of some clues that we're dealing with X-linked dominant traits. So these conditions are expressed in the female when just 1 copy is present. So we're dealing with a mutation that's carried on or associated with a dominant allele. So to denote that, we use our X because it's X-linked. And in this case, we're going to use the capital letter. And for mutation, I'm going to use the M. So as long as the female gets 1 copy of this mutated allele, it's going to be expressed. And that's because it's dominant. Therefore, we require just 1 copy because it's sufficient to mask whatever normal copy we see on a recessive allele. The only thing to keep in mind is that if the female, even though it's possible she only has 1 copy, she will show symptoms associated with the disorder. There are some cases where a heterozygous mother may not show symptoms or may have mild symptoms, and that's due to X inactivation, which again we'll get to in a few slides. So these types of conditions have much more severe effects in males. Sometimes they're so severe, in fact, that we don't even see males survive to be born. So really high rates of miscarriage due to early lethality. And that's just because these alleles are, the mutation is carried on a dominant allele. And remember, those are typically gain of function effects. So we have the presence of a new enzyme and one that is completely alters the function of the original protein. So extremely severe effects. These conditions are also passed from males. So here we can see if we do have an affected male, so he survives to be born, and the condition in some cases isn't severe enough to where they pass on the mutation, the male will pass the condition on to every single one of his daughters. That mutation is going to be passed on, but not to any sons. And that's because he's only going to pass on the Y chromosome. So an X-linked dominant condition that is actually mild enough to be passed on to children, so we can see examples of it in pedigrees, is a condition called incontinentia pigmenti. So in this condition, we see swirls of skin pigment that arise where we have melanin which is simply a type of cell that provides coloration, penetrating the deeper skin layers. So melanin we typically see in higher up within the epidermis, but in this case it's penetrating down into the dermis. So this presents as even yellow pus-filled vesicles on the limbs, can kind of come and go, but we have lesions that remain throughout life, even if they fade with time. So this is something that's not going to affect the normal functioning of the body or any really important processes, but is showing kind of these superficial issues. So when we look at the Punnett square, as I mentioned, these types of conditions are not typically passed on via the male side, and that's because they can often be more severe for a male. So in this case, we're going to have an unaffected male here on this end of our Punnett square, and we're going to have an affected heterozygous female. So she only has one copy of the mutation. So this affected heterozygous female, half of her oocytes are going to carry, I like to use green to show the mutation, half of her oocytes are going to carry that dominant mutant, and the other half are going to be normal wild type. And for our male, he's going to pass on the normal wild type allele and a Y to his sons. Okay, so here we see in the Punnett square that 50% of the daughters in this scenario are going to be affected, and 50% are going to be unaffected. So remember, with our X-linked recessive traits, we saw no females affected unless dealing with X inactivation. But without that, we didn't see, think about that pedigree of hemophilia. We did not see affected females. Now in this scenario, we've got a 50% chance that the daughter will be affected. And that's because she's inheriting the mutant dominant allele from her mother, and the condition is dominant, so one copy is enough for the symptoms or phenotypes to present. So we look down at the sons, and it's exactly the same as our previous scenario. So we have 50% of the sons are going to be affected. This is always passed from the mother, and 50% unaffected. But the key difference here is that there are some cases where males who inherit that mutant allele may not even survive to be born. So with this condition, when a mother has the disease, as we see in this Punnett square, she actually has a miscarriage rate of about 25% if that developing embryo were to be male. So now we've covered inheritance patterns involving X-linked recessive and X-linked dominant disorders. So we're going to add that to the list of all of these other conditions that we've been looking at. So how do you use this information to solve genetic problems? And this list of stuff actually applies to any of the questions that I may present to you on the quiz or the upcoming exam. So the first steps that we would follow, and the first three actually are taken care of because I like to give enough information for you to start working on the problem. So the steps that we would take if we didn't have any information at all, when you're a genetic counselor and you're working through you're trying to determine what the condition actually is that you're studying. So you take a look at the inheritance pattern, and to do that, you can even draw out a pedigree. So sometimes we're not presented with a pedigree, but in other cases we are, and you can track the affected and unaffected individuals. So once you have your pedigree, you can list out your genotypes and phenotypes with their probabilities. So knowing what you know about the condition in the wider population. So I'm going to give you all that background information. So the steps that you're going to need to follow is then assign genotypes and phenotypes to parents. So I may give you half of the information, but your job will be think about how the pedigree looks and then what you think the associated genotypes and phenotypes of the parents would be. And then, as we've done in the previous two examples where I walked through Punnett squares with you, once you know the parents' genotypes and phenotypes, you can determine how the alleles are then going to be separated into the gametes and passed on to the offspring. So using that Punnett square to determine, hey, what are our ratios in terms of genotypic and phenotypic ratios? And repeat that for the next generations. So once you go through this enough times, you'll get clues as to whether the condition is manifesting as an X-linked dominant, an X-linked recessive, or even for other examples that we've covered so far. So we're now going to cover an additional 2 types of traits where we see gene expression and therefore inheritance patterns differ between males and females. But we're not going to deal with the X-linked dominant or recessive scenarios that we just covered. So sex-limited traits. So these are traits that affect a structure or a function that occurs only in one sex. So the phenotypes are limited to just one sex or the other. So the gene, the underlying gene that contributes to the phenotype, and this is important, it can be either autosomal or X-linked. So it can be on any type of chromosome, but the key is that the protein that's produced from this gene is actually going to influence either the structure or the function that we see in just one of the sexes. So some great examples of these. One is beard growth. So if you have a gene, let's say, that causes for the production of hormones related to beard growth, we know that beards are limited to males. So in that case, we are dealing with a gene that affects kind of the structure of this male phenotype. So it's sex limited. Another example is milk production. So milk production is limited to females because they're the individuals that have the hormones and the structures that allow for this production. So because it's only occurring within one of the sexes, again, sex limited. Finally, another example is preeclampsia. So that's a sudden rise in blood pressure that occurs late in pregnancy. It actually kills about 50,000 women worldwide each year. But what we know is that because preeclampsia is only occurs associated with pregnancy, which only happens for the female sex, again, this condition is sex limited. So not to be confused with sex-limited traits, we also have traits that are sex-influenced. So these are traits in which the phenotype and this is the key, expressed by a heterozygote, is influenced by sex. So what this means is that the allele underlying a particular condition is dominant in one sex, but then recessive in the other. Okay, so we don't have an allele being always dominant or always recessive. It's one for one sex and then the other in the other sex. So same deal, this can actually occur on either the X chromosome or an autosome, it doesn't matter. But a great example of this is pattern baldness in humans. So a male who is heterozygous for the condition is actually bald, but a female is not. So what does that mean? The male has just one copy of the mutation. So he's heterozygous. But he shows the condition, shows the associated phenotype. So that means just one mutant allele is enough to show the phenotype, so it's dominant. However, for females, a heterozygous female is not bald. So what that means is that she would need two copies for the associated phenotype to be expressed. She has a functioning copy that masks the effect of the recessive allele. So in this case, baldness is recessive. So you need to look for keys in the scenarios to determine how you have differences in allele expression between the sexes in these types of scenarios. So typically what happens is you have one sex that is more likely to have the condition than the other, because for one it's dominant and for one it's recessive. Another example when I'm setting up these scenarios is when I describe that two individuals have the same exact genotype, but one shows the condition and the other doesn't. So now I want to explore this topic that I've referred to a couple of times, X inactivation. So remember, females have two alleles for all of their X chromosome genes, just like the rest of their autosomes. But males only have one. And that's because females have two copies of the X chromosome. They're the homogametic sex, and males are heterogametic. They only have one copy. Some we refer to them also as hemizygous. So this kind of sets the stage for some inequality with regards of total number of allele copies. Females would have twice as many as men. So to balance this inequality in mammals, X inactivation plays a role. And X inactivation does exactly what it sounds like. One X chromosome is randomly inactivated in each cell. So because of that random nature, the one inherited from the mother or the X inherited from the father can be inactivated. So because of this, a female mammal can express the X chromosome genes inherited from her father in some of her cells, but can also express the X chromosome genes inherited from her mother in others. So we typically refer to women as a mosaic for expression of most of the genes on the X chromosome. And the X chromosome that is inactivated in each cell is referred to as a Barr body. So this process of random inactivation of one of the X chromosomes in each of the body cells actually occurs very early on in prenatal development. And on the next slide, I show a schematic for how this works from development progressing onwards. Because one X chromosome is shut off in each of the cells, we can have alterations to our phenotype. And we refer to this as an example of an epigenetic change. So that means that there's an inherited change. It's not altering the DNA base sequence. These changes aren't due to a mutation, but they're due to simply which alleles are being expressed. So a great example is if we have a heterozygous female, we have a condition that's X-linked recessive. So she should not show any symptoms associated with the phenotype. But if randomly that normal wild type allele is the one that's shut off, the recessive allele is the only one that can be expressed. So she can show symptoms associated with the condition. That phenotype is altered, but the underlying DNA sequence is not. So the gene exist encodes an RNA that actually binds to a specific site on an inactivated X chromosome. And what this does is, from that location where it binds all the way to the chromosome tip, most of that X chromosome is then silenced. So that's at the molecular level. How this actually works is that we have a gene that encodes an RNA to ultimately bind and activate one of the X's. So how this works is we have a fertilized ovum at the very beginning. So this ovum has 2 copies of the X chromosome, because that's what we start with, a maternal copy and a paternal copy. And then remember, as we're going through early development, we're going to go through many rounds of mitosis. And what you can see is that early on, this row of cells where X chromosome inactivation actually starts to progress, each of these cells are random with regards to which copy of the X is passed on to the next generation of cells. So one X chromosome remains active, the other is inactivated by that exist gene, and it becomes a bar body. So there's no longer any expression. So these cells then go on to give rise to the next generation of cells and the next generation of cells that we see all over in the body's tissues. And so you can see how, if we're looking at skin because it's a large organ, the largest organ of the body, how it has so many cells that comprise it, but each of those cells are random with regards to whether the maternal or paternal X chromosome is present. So you can see how this kind of gives rise to this mosaic pattern. So in this case, if we were to say that the green cells that are carrying only the paternal X copy have a mutation associated with them that normally would be recessive, we actually see the expression of that recessive phenotype on parts of the female body because some of those cells just randomly are showing the recessive allele activated. So another way to refer to this mosaic nature of expression of X chromosomes is to refer to a female that expresses a certain phenotype that normally would be associated with a homozygous genotype as a manifesting heterozygote. So the consequences of this X inactivation on phenotype can be really interesting, can also make things really complicated when we're looking at the inheritance of different phenotypes or disorders. So obviously, if an individual is homozygous for an X-linked genotype, they have 2 of the same alleles on their X chromosomes, inactivation is going to have no effect because it doesn't matter whichever of the X's are inactivated, we still have that same allele on the other X. However, for heterozygote individuals, they have two different alleles. But X inactivation is going to lead to the expression of one allele or the other. And so that's why we see all of these cells as a mosaic nature. And sometimes, because we have one or the other, it doesn't affect the health if enough cells are actually expressing the functional product. So if randomly the majority of cells are expressing the wild or normal functioning allele, we can have a mother who has mild symptoms associated with the disorder, or even no symptoms at all. On the other hand, if randomly the mutant alleles are the ones that end up being passed on and maintained over the cell's generations, that mother could actually show a lot of problems associated with the condition. A great way to physically see this representation of a mosaic phenotype is to look at calico cats. So all calico cats are females, and the fur coloration is due to a gene carried on the X chromosome. And so it all depends on the allele that is the one that remains activated with regards to the different coloration of the patches of fur. So another factor that can influence gene expression in different ways between males and females is genomic imprinting. And so this refers to when the phenotype of an individual actually depends on whether the gene was inherited from the mother or the father, so that parental origin. So genes are actually imprinted by an epigenetic event. So remember, epigenetic means that there's a change in a phenotype, but not a change to the underlying genotype. So it's passed on from cell to cell, but not from individual to offspring. So how this works is that there are methyl groups that cover a gene or several linked genes, so a whole sequence of DNA. And because these groups are covering the DNA, they actually prevent them from being transcribed or translated. So we don't see the proteins being produced if the methyl groups are bound to a region of DNA. And this is all in a pattern determined by the individual's sex. So, these imprinted genes, one of the copies inherited from either the father or the mother is always covered with methyls. So we could have a gene, for example, that would function if it comes from the father, but it won't function if it comes from the mother because that's where the imprinted gene would be methylated. So it's a parental effect on gene expression, and it's seen in disorders that are always inherited from either the mother or the father. That's your clue that you're dealing with genomic imprinting. So even though we see this imprinted gene being passed on to generations of cells, we don't see the imprints being passed on to the gametes. And that's because even though the DNA may be silenced for expression, when it's replicated prior to meiosis, all of those imprints are removed and reset. So the methyl group shielding the imprinted genes are stripped away, new patterns are set down, and the offspring can look very different from the parent. So the function of this isn't very well understood, but many of these imprinted genes actually take part in early development and key proteins in the embryo. So even though we don't completely understand the function of genomic imprinting, research suggests that it can contribute to requiring two opposite sex parents to produce a healthy embryo. And that's because we have genes being covered with methyls depending on which parent the homolog came from. And interestingly, the male genome controls placental development, but aspects of the female genome controls embryo development. So there are components from each genome that contribute to different aspects of development. It can also explain incomplete penetrance. So remember, that refers to the all or none expression of a phenotype. So when individuals can have the same genotype, but someone has the condition and someone else doesn't. So the penetrance of the genes within the phenotype that we see is incomplete. And this can explain how this works molecularly. The number of imprinted genes in the human genome exceeds 156, and at least 60 of those affect health when they're abnormally expressed. So the effects of genomic imprinting are only noticeable when an individual has one copy of a normally imprinted allele, so that means it's normal for it to be silenced, but the other allele copy, which should be active, is somehow inactivated or completely deleted. And these types of diseases can have dramatic symptoms associated with them, and only due to just small deletions of DNA. So your book covers two distinct syndromes that result from the very same small deletion of the same region of chromosome number 15. Just depends whether that deletion is inherited from the father or from the mother. So Prader-Willi syndrome, which presents itself as difficulty in gaining weight and growth early on in development, but later, as a child is in the range between 1 and 3, they obtain an obsession with eating, but very slow metabolism. So these children can actually eat themselves to death because they don't have any mechanism to shut down hunger. And this syndrome actually arises because of several paternal genes, so this is inherited from the father, that are not normally imprinted. So that means they should be normally active, but they're missing. So that deletion is passed down from the father. Normally should be expressed, but it's not. In Angelman syndrome, which is a syndrome that causes autism, intellectual disability, problems associated with the musculature in the face and arms, same deletion, but it's inherited from the mother. So a normally active maternal gene, it's the one that should be expressed, but it's deleted, it's missing. So this particular region that we see being affected by a mutation on chromosome 15 is actually extremely unstable. So that's the reason for this being common and common in both sexes. And that's because the DNA sequences that are on either end of the gene have high repetitive segments. So we've talked about DNA that has lots of repeated bases are more prone to errors, and that's the case with these two conditions. These two syndromes may also be due to uniparental disomy, and what that means is that for whatever reason there's an error with meiosis, non-disjunction occurs, and so extra copies of the chromosomes are passed on. And in this case, you end up inheriting two of the same homolog from one of your parents rather than one from each.